Welcome to Exam Mini Video 6. Today we are going to be talking about developmental networks and what happens when mutations occur within those networks. You will better understand this topic if you know these terms. Please pause if any seem unfamiliar. So what is a developmental network? A developmental network is a sequence of genes whose individual protein products have activating or repressing effects on subsequent genes in the sequence. To understand how this works, let's look at a network using characters from the TV show Criminal Minds. Within the network, arrows represent activating relationships and T-bars represent repressing relationships. When all the genes are wild type, rendering them functional, the genes interact to produce a normal end phenotype. In this case, SSA Rossi, one of the founders of the BAU, activates SSA Prentice, the team leader of the BAU, who is then able to repress the hitman who is hired to take out or repress the BAU's technical analyst, Penelope Garcia, but because he is repressed, he cannot repress Garcia, so Garcia is functional and is able to provide information to the team Brainiac, activating Dr. Spencer Reed, who uses his super smart brain to solve the case, and suppress the end sub, resulting in the end phenotype of apprehending the criminal. Let's look into the biology of how this works by focusing on a couple of individual relationships. In an activating relationship, such as that between Rossi and Prentice, the Rossi gene is able to transcribe Rossi mRNA, which is then translated to produce the Rossi protein. That protein then binds to the Prentice gene, which promotes the transcription of Prentice mRNA, and consequently, the translation of the Prentice protein. In a repressing relationship, such as that between Prentice and the hitman, the Prentice gene is able to transcribe Prentice mRNA, which is then translated to produce the Prentice protein. That protein then binds to the hitman gene, which ultimately inhibits the translation of the hitman mRNA. But what would happen if we had a single gene mutation within this network, say in the Prentice gene? While the Rossi gene can still activate the Prentice gene, it does not matter because the Prentice gene is non-functional, so inhibition of the hitman gene is not occurring. Now, the hitman gene is functional, so inhibition of the Garcia gene is occurring. Now the Garcia gene is non-functional, so activation of the Reed gene is not occurring. The Reed gene then is non-functional, so inhibition of the killer gene is not occurring. And our end phenotype is an unapprehended killer on the loose. What would happen if there was a double mutation in the sequence so that both the Prentice and hitman gene were mutated? Again, the Rossi gene's ability to activate the Prentice gene has no effect, because the Prentice gene is mutated and non-functional. So again, repression of the hitman gene is not occurring, but this time, because of the second mutation on the hitman gene will not be allowed to function properly. The repression of the Garcia gene is not occurring. The Garcia gene is therefore functional and can activate the Reed gene, so now a functional Reed gene can successfully repress the killer gene, and we get the end phenotype of an inactivated and apprehended killer. That is the end of this video. Stay tuned for the final one of the series in 2 weeks.